developed a method in our lab, and based on genotyping by sequencing, extending it with bisulfite treatment, and other people have done the same for red-seq, extended with a bisulfite treatment. And because you only look at small snippets of the genome, you actually do not need a reference genome, because from your own data, you can reconstruct the reference of the small parts of the genome that you have in your data set. So this suddenly opens up DNA methylation analysis also in species for which you do not have a reference genome, which is really nice and cool, and we're all excited about that. But we have to keep in mind, you're looking maybe not even at 1% of the genome. And the additional problem, if you don't have a good reference, if you do reduced representation bisulfite sequencing with a reference, you can align everything, and you actually know what regions you're looking at. If you have to reconstruct the reference, you only know what regions you're looking at if these snippets hit a gene that can be annotated using information from related species that are out there and that you have the information on. So there's only small parts of the whole genome that you can annotate in principle if you take my dandelions, for instance. And so these are really the limitations. You can look at DNA methylation in the genome, but are you looking at what regions are you looking? Are you looking at the DNA methylation that's functionally relevant? All these things we don't know with RRBS methods and non-model species. So to quickly summarize the pros and cons, so what can you do and what can you not do? So if you compare whole genome bisulfite sequencing to reduced representation bisulfite sequencing with or without a reference genome, and so I think one thing is clear: you're completely fine. For instance, if you look at the methylation information in terms of, if you think in terms of marker studies, so you do not need to look at the whole genome to characterize patterns of epigenetic variation within populations and between populations, or how overall profiles epigenetic profiles correlate with environmental conditions. And so all these things, I think, in terms of using the information as you would do for markers, but then epigenetic markers and not genetic markers. You only need, you could do it with whole genome bisulfite sequencing, but really, you don't, if you have several thousands of sites in the genome, you have enough for all kinds of descriptions of epigenetic diversity in the system. But for functional analysis, of course, you can do almost everything that you want here, and RRBS is also really good if you apply it to a species with a reference. Without a reference, you can also do something, but it's quite limited what you can do. Then, I have to add that this is particularly limited in the case of plants. So if you work in animals, at least in mammals and many other animals, there's a lot of functional DNA methylation variation sitting in what they call CG islands in the promoters of genes. So methylation variation in CG islands in animals is associated with function. And you can trick, depending on which enzyme you use, you can trick your RRBS methods to bias strongly towards CG-rich areas. So in many animal species, the opportunities for functional analysis with RRBS are much higher than in plants. Plants lack CG islands in the promoters. So that leaves you with quite some limitations in the functional analysis. But as I said, if you work on animals, which some of you do, there's actually these methods give you more power for functional analysis. But still, we applied this method. A PhD student in my lab applied one of them, epiGBS we called it, so genotyping by sequencing, but then with an added bisulfite treatment. It's relatively cheap, I would say really cheap. So we've done old-fashioned methylation-sensitive AFLPs previously. It doesn't differ that much, I think, in terms of costs. But this is one experiment that we did, and it gives an illustration of the type of interpretation that you can do, even without going into really functional analysis. So, for instance, one thing that we did is only looking at the control plants. You could simply see what is the dynamics of DNA methylation across generations within the same genome. So like these Arabidopsis mutation accumulation lines, then for 6 generations. And we saw that the overall genomic correlation in CG methylation levels across a genome. It's really, really high, even if you're 6 generations removed. So correlation between 0.95 and 0.98. So this means that this means what we actually know already from other plants, that CG methylation is really, really stable. So it goes from 6 generations removed. It's almost the identical methylation profile in CG context than 6 generations ago. Part of this is because CG methylation may be under genetic control, so if it's lost, it can be rebuilt again in the same way in every generation. But we know, not from this study but from other studies, that CG methylation actually in plants, part of it is not even reset between generations. It's just six there. So the more interesting thing, we didn't know this about CG methylation, but to some extent we see the same pattern for CHG methylation, meaning methylated cytosines, but not in CG context, but C and then another nucleotide and then a G. So this is a lot you have this a lot in plants and you have this less in animals. But you see a similar pattern, so the correlations are much lower. This is much more dynamic, and also we can measure it with less accuracy, because this CG methylation, it's often either close to 0% or close to 100% in a sample, really methylated in all cells in your sample or not. This can be like 30% methylation or 35% methylation in a given sample, and the lower these numbers get, the less accurate we can estimate them from our data. But you see here a same trend, that plants that are removed from one generation have a certain correlation in the CHG methylation, and the correlation generally decreases with increasing generational distance. So this is divergence building up over generations, meaning that CHG methylation has quite some dynamics. There's variants arising, and moreover, at least a component of those variants are stably inherited. Because if they would not be inherited, you would not get gradual divergence with successive generations. So this is actually good evidence, even though we don't have this is not a functional analysis, but it is good evidence that not only CG, but also CHG methylation in these plants has a component that is stably inherited. So the other question is, of course, what is the effect? Do we see an effect of this environmental treatment on overall patterns of DNA methylation in the genome? And the answer is mixed. The answer is yes and no. We did this in two different, the whole experiment, two different apomictic genotypes. So apomix that's what this form of asexual reproduction in plants is called. Animal people would call this parthenogenetic reproduction. Apomixis means clonal seed production. So in one genotype we found nothing. In another genotype we found that if we take actually not these plants but the offspring of these plants all grouped together compared to the offspring of these plants all grouped together, then we see that we do not see an effect that these guys are different from these guys if we look at methylation in CG context. But we find a strong effect if we look at the DNA methylation sites in CHG context. So that suggests that the environment-sensitive component in DNA methylation is visible in CHG methylation here and not in CG methylation. So the environment-induced component, that is then inherited for at least one generation, because again, we did not measure these plants themselves. We measured the offspring of all these plants when we raised them after the experiment in one common environment. So, and a bit more detail, this result split out for the CHG methylation in this particular genotype. If we simply do not compare everything, the whole group, but if we do this by generation, we see that the differences build up. You need, let's say, two generations of parental exposure before the offspring shows a significant difference. And here it's also, well, it's not significant, but there's at least a relatively low p-value. Here there's still a difference. And the offspring of these guys are not different anymore from the offspring of these guys. So this is not completely unlike what we've seen what other people have seen previously in Arabidopsis. So what it means is that if you trigger a plant with a stress, jasmonic acid in this case, we see inherited effects on gene expression, on leaf chemistry, and we see that it's also, at least in some genotypes, we see it in DNA methylation, and we may still see a small hint here, but after that it dies out again. So with that, I think the take-home message that I see from this type of work is that, first off, I think asexual lineages are really cool models for studying epigenetic inheritance and the role of epigenetics in ecology and evolution. And we've also seen that rapid divergence within an asexual plant lineage, in flowering time in this case, is at least mediated by DNA methylation. And we don't know yet at this point if DNA methylation differences are the ultimate causal factor in determining it, or that they are really part of, let's say, the flowering time phenotype, that there's some genetic determinants causing the DNA methylation determinants which cause the flowering time variation. So this is for future work. And we see that all these studies that we did, we see that part of the environmental response in these asexual plants is sustained into offspring generations, but it's not sustained. It sticks for part of it sticks for one generation, a tiny part maybe for 2 generations. Then we cannot detect it anymore, which makes perfect sense, I think. And just to wrap up, if I think about the future prospects of this type of work, so with this type of work, I mean epigenetics in basically non-model plant species or not in model species. So we're interested in the diversity and adaptation that we see in the wild, and yet we base almost everything we know in plant epigenetics on what people have seen in Arabidopsis, this tiny plant with a very short lifespan. And I think the next step really is to good high-resolution epigenetic research, ideally like what you can do in Arabidopsis, but apply it to the richness of species where epigenetics can play very different roles. For instance, as I highlighted, I think epigenetics can be very different in asexual species compared to sexual species. I will predict that it has a different role in long-lived species compared to really short-lived annuals like Arabidopsis. You can also hypothesize that the role is very different in species that occur in very dynamic habitats versus species occurring in very stable habitats. So what needs to be done, I think, is to extend the high resolution. The epigenetic research has to become high resolution because of the nuances of the underlying epigenetic mechanisms that you simply need to be able to look at. But we want to apply that to let's say, a range of different species with different traits. And with that idea, we have a European training network in which we try to actually do that. So we got together with ecologists, molecular biologists, and some bioinformaticians with the goal to execute a common research program applying high-resolution epigenetic analysis to a couple of very different natural plant species. So we just started that. There's 15 PhD students in this network, and we're studying Fragaria, which is a wild strawberry that can reproduce clonally and sexually. We study a tree, Populus nigra, and there's not a species involved. So the next move, I think, in the field is to really try to do high-quality epigenetic analysis, but apply it to natural systems. So also the dandelion system, I think what I showed you, I think these are nice initial experiments, and this is what you can do if you do epigenetics in a species for which you don't have a genome. But you get to here, and the questions that are now on the table require more detailed molecular and genomic work. So we in fact generated the reference sequence of dandelion now with some other groups. So that luckily is also not outside of reach for many groups now. So it's become relatively inexpensive to do that. And I honestly think that this is the way to go if you want to do epigenetic analysis in natural systems and go beyond getting first glimpses of what's going on. And with that, I'm left with thanking the people who helped with the experiments that I showed. So these are people from the group that helped over the years at the Netherlands Institute of Ecology, particularly Thomas van Gerp. He's a PhD student who developed this epiGBS method that we use now. And Christian and Morgan are recently started PhD students in this European network. The guy, the dandelion expert who could recognize the samples from an individual clonal lineage in the field, Jan Kirchner. people in the University of Florida who helped with the RNA study, and in Tübingen who helped with the metabolomics, and in Nijmegen as well. And with that, I'd like to thank you, and I'm happy to take questions.
Interesting and very stimulating, I think. So if there are questions from the audience, please don't be shy.
Just a curiosity, if the flowering time was synchronized for the latitude?
Not really. There was a trend. No, actually, there was not a trend. So the accessions came from different latitudes, and the accession is different, but it was not correlated very well with the latitude. So to what extent it is actually an adaptation, we don't know. It may just be variation that has developed that is not really adaptively selected. Then again, we cannot really tell. We know from other species, again, Arabidopsis and studies that people have done there that, yeah, sure, there are some large geographic trends in flowering time, but superimposed on that are micro-habitat patterns that select for specific flowering times that can overwhelm the large-scale geographic gradients. So what we look at here is in the dandelion system, the flowering time variation itself was not correlated with the geographical gradient, and whether or not that means that it's not adaptive, I can't really tell. I'm not sure.
I remember from the 1980s in the Netherlands that people in Wageningen wanted to recognize, instead of one dandelion species, literally tens and tens of dandelion species that showed morphological differences and that did grow in different environments. Is this the explanation for all that difference?
No, this is not the explanation. So one part of the answer is that in these asexual systems, partly asexual systems, the species concept you can debate what is a species then at some point. And I know that in this one particular species, Taraxacum officinale, it's called, that you have your sexual plants, diploids, and you have this triploids, asexual plants, but you have thousands of them. And some people call those each lineage can be called a microspecies, or a cluster of lineage can be called a microspecies if they come from the same genomic background. So to me, this is semantics. I don't know what is the right term here. But for sure, there's different genotypes even within the species. And for sure, the different genotypes, or the different clonal lineage, have different traits. So if you look closely enough, you can find these. So these differences, they may not have to do anything. They may or may not have something to do with epigenetics, but you don't need epigenetics to explain the differences between different clonal lineages.
Thank you for this very stimulating seminar. And indeed, the microalgae with their high generation time and also the switch from clonality to sexual are ideal, to me, would be ideal organisms on which to apply these techniques and understand better. And my question is still related to the flowering time, as it has been considered as divergence in flowering time as very important for speciation and species divergence, because you have then this early flowering and late flowering that reinforce their differences by sexual reproduction. And so my curiosity is, what is the next step after methylation? So I can figure out that that epigenetics, this mechanism create a lot of diversity and a lot of opportunity for a species to colonize different time frames and speciate eventually. But when the switch from one modality, that is that the flowering time is modulated by this methylation, and the next step when it is really genetic, how do you think it can happen?
So I don't know if I scroll back quickly to the. Maybe I I should show this slide. Well, first this slide, so that I just showed. So I think, so the next step, the interesting the question that I'm interested in with this flowering time result, you have flowering time differences, and we make the differences go away if we make the DNA methylation differences go away. So the question that I'm really interested in is whether we're looking here at DNA methylation variation that is the ultimate cause, the ultimate and only cause of the flowering variation, or whether the methylation variation itself is actually part of, let's say, the phenotype, and there really is a genetic difference between the plants that then causes the methylation differences, and which then cause the flowering time differences. So to me, in my mind, there's a causal arrow. If we believe these results, there's a causal arrow going from methylation variation to flowering time variation, but we don't know. There could be an unseen causal arrow going from genetic variation to methylation variation, and this is what I try to explain here. So this could be a possible explanation for the genetic link. So here. So these could be two different plants, two different accessions with difference in flowering time, and maybe this is a flowering time gene. And it could be that the genetic cause, the ultimate genetic cause of everything that we see, is a transposable element insert that is present in this plant but not in this plant. And because of the DNA methylation effect, silencing of the novel insert, maybe also there's DNA methylation silencing of this flowering time gene. So if this is the reality, then I would look at this system as ultimately caused by genetic differences, and the genetic differences cause methylation differences, and the methylation differences cause the flowering time differences. So from a conservative perspective on epigenetics, would propose this as a logical hypothesis of what is going on. I would be interested in finding this out, because it doesn't have to be like this. We've seen that epigenetic variants can also simply arise spontaneously as epigenetic mutations that are even in the exact same genetic background. And if that happens, and if then they are transmitted stably cross-generation, then you could have these flowering time differences without any DNA sequences differences. So the question here really is whether the DNA methylation, whether you should look at it as part of the phenotype, whether it's a phenotype, or whether you should think of it as a genotype. And to me, that is in this particular case, that's a relevant question. And to answer that question, we need more genomic resolution to be able to look at the relevant genes.
I was wondering, when you talked about your predation, the gismonic acid, okay, so, and the fact that it disappears very quickly. Have you tried with other environmental factors like, just thinking, like a very hot summer or drought, or do you expect that depending on the stress you would see the effect over a longer time scale? Is there any evidence of this?
Yeah, so there's an adaptationist answer to this and a non-adaptationist answer to this. So it depends on whether you think the processes that we look at are in fact evolved adaptations and they make sense, and the inheritance of them makes sense exactly because what you say. So people have done some theoretical and modeling work where they showed that it depends on the dynamics of the fluctuation of the environment, the scale of the fluctuations, whether or not these inherited plasticity is adaptive, yes or no. And I think they showed that if the stability over generations, the level of stability matches the level of the fluctuations in the environment, if that happens, then yes, that is the transgenerational stability can be selected for. It can be adaptive. And then your question translates to the question, what environments fluctuate at the scale of oscillations over multiple generations. So for instance, I'm fantasizing here, but let's say certain herbivore or pathogen outbreaks that stick for a couple of years and then go back again. So if that is the environment, then in such environments, there can in fact be selection for the transgenerational stability of the induced responses. So that would be the adaptationist answer to your question. And then I would agree. I would say yes, if we find an environment that fluctuates at a level that is relatively slow compared to the generation time of your organism, then you would expect that the stability of the induced effects depend on the stress. Then maybe if it is drought that shows these oscillations, then you would expect the drought effect to persist over the number of generations. Of course, at this stage, we don't know if what we see is adaptive. That's the other answer. So it could be that we're simply looking at some side effects of the mechanism that could not be avoided, or that the plants would like to reset if they could, but for some reason it doesn't. At this stage, we don't have good evidence that the pattern, the things that we see, are in fact adaptive. Having said that, I'm thinking of one experiment that I know from C. elegans, the worms. So the worms can be learned to be attracted or deterred by certain chemicals in the environment. And somebody has shown that if you expose worms for multiple generations to a new compound that they're associated to become attracted to, they've learned that preference for that compound sticks for the 40 generations or so that they perform this experiment. So you can also hear they didn't show that this was an adaptive benefit, but you can easily imagine how something like that could have an adaptive advantage. That didn't work through DNA methylation, by the way, but through another epigenetic mechanism.
Okay, somehow related to that. If I correctly understood, methylations are linked to, let's say, either stress or changes in the environment. And what is known concerning the disappearance of this methylation? Is it random, or it can be also linked to something in the environment?
Yeah, I think here I simply don't know, but that's my lack of expertise in that part, I think. So there are specific enzymes that are designed to specifically remove DNA methylation from the genome. So there's, to some extent, there's an active process, possibly then also a targeted process in removing DNA methylation. So DNA methylation can also be removed passively by simply reducing the activity of the enzymes that are necessary to maintain methylation across cell divisions. So that would be, let's say, a passive removal of DNA methylation. That occurs, for instance, in germlines and during early embryonic development, to erase DNA methylation from the genome and then rebuild it again. And again, how this works is different between plants and animals. In plants, I also know that there's other active removal of DNA methylation that is not targeted. There's an enzyme that also removes it, but I think it removes it across the board, and I don't think it has specificity. But it's my lack of knowledge. I would guess that there's enzymes that also have an active, targeted removal of DNA methylation, but I'm not the right person to ask this to.
But we have some initial evidence that stressed plants accumulate methylation more than control plants. So that stress induces methylation, and also the different parts of the same plant accumulate different level of methylation, young tissue respect to old tissue, and so on.
You ask if this happens? Or
No, I say that we have this evidence.
Oh, I think that's really interesting. We were in fact starting to work on this in trees. We were thinking along the same lines that it would be nice in a long-lived individual to see how an individual can be an epigenetic mosaic to some extent.
Yeah, we have some evidence.
So that would be really interesting to talk about. Yeah.
Can I destroy this part? We have different evidence. Indeed, but it is interesting to compare also the approach, probably, because in stress we have under methylation, and we have expression of transposable elements. Yeah. So indeed, the feeling that I had, and we can discuss on that, is that in that case, is a maintenance that goes out, is not under control during the stress. But getting to the, I have several questions, but I will postpone them. But because you stress the part of the technological approach.
Yes.
How would you deal in this case in which you can have changes at micro scale in the genomes with the difference in methylation? Because indeed you cannot see this by bisulfite treatment because you have changes at the sequence level.
Yeah. So I think any approach here will be helped by a good quality reference genome, but this particular question, you could in fact also, well, you need some reference sequence, but it could be part of the transcriptome that is easy to obtain. So no, I step back. I think what I would do now, we have the reference genome, we know there's many candidate loci for flowering time in plants. So we've actually repeated I didn't show, but we repeated the zebularine treatment, but then measured at gene expression level. So we identified 9 or 8 candidate genes from different corners of the flowering time pathway that we know from Arabidopsis. So we looked for the respective genes in the dandelion genome, and we did gene expression. No, we try to zoom in which corner of the pathway something seems to be happening with, where the expression of a gene is also sensitive to this demethylation treatment. So we try to zoom in a bit. So we have a few genes that show an interesting expression level, interesting differences in expression, and having those candidate genes. But you could also simply, take candidate genes that you don't have prior information on in your own system. So you could do, in this case, we would probably do PCR amplification with bisulfite treatment to see from, maybe one end, see if we can design primers, the 5' end of the gene extending into the flanking region. And of course, this then will be helped with having the reference. And then simply compare different accessions, whether there's different transposable elements inserts that show up.
Okay, I think we can free Kuhn. Thanks a lot for the seminar. This was very interesting. And, you will be here today in the afternoon. We have a meeting in the lab, but if you're interested, just contact me if you're interested to talk with me.